I can guarantee you, in this short video, there is absolutely no way I can give you the whole picture of what goes on at the chromosomal level of determining hereditary traits. This is one where you will definitely need to go do some supplemental readings and do a little bit of searching yourself to fill in. But right now, mostly I want to give you some definitions and maybe a little review for you if you've already had this before of what is going on in terms of physical development that you might say we have no control over, it's all genetic. We're talking about inherited traits. Traits that you do not have any control over. It's all been decided at conception, basically. You have to remember that the genetic code for each individual is on chromosome pairs. Now, egg and sperm have half chromosomes that match up to create a whole pair. And that sex-determining chromosome pair would either look like XX, female, or XY, male. And how do we know which one we're going to get? The sperm determines it because the sperm is what has the Y. It also has X. So the sperm is going to determine the sex of the child. The X chromosome carries on it particular specific female traits. The Y has very specific male traits on it. But on other chromosome pairs, which trait you get is determined by which is the dominant gene of that pair of the two. And there are times when traits need to be analyzed at the chromosomal level. And so we have to make sure we understand our vocabulary here. The sex chromosome is the allosomal chromosome. And all the others are called autosomal. An example. If a person has red-green color blindness, is that because of something allosomal or autosomal? Well, it turns out that color blindness is not sex-linked. Therefore, we would say it is autosomal. It is determined by chromosomes that are not the sex chromosome. Sometimes, though, it's a lot more serious, what we're trying to analyze or what we're trying to learn, a lot more serious than just hair color or color blindness. And so there is a process known as amniocentesis that is used to find if there are going to be genetic anomalies on chromosomes that will end up manifesting in physical appearance or physical behavior. Amniocentesis is done during the 14th to 16th week of pregnancy. Amniotic fluid from the womb is drawn out so that it can be taken to a lab and analyzed by its genetic code. The chromosomes can be studied, and certain markers can be looked at specifically that can identify particular and well-known genetic flaws and abnormalities that are associated with those particular markers. For instance, Down syndrome can be determined before birth by using this process. There's sometimes, rarely, but sometimes a blood type mismatch between fetus and child. It can be found through this process. And such things as neural tube defects. As the neural tube is being developed in the fetus, it can be analyzed through this process so that we can determine, is this child going to have spina bifida, for instance? There are other kinds of things that can be determined by amniocentesis, and you might want to do some extra research to learn about this some more. As I say, this was just an introduction, and to make sure you got all the vocabulary words that you're going to need for understanding and showing that you understand all these kinds of processes.